I'm aware that you might be waiting for lunch as much as I do, so I'm going to keep within my 15 minutes. But yeah, I'm going to switch gears now and speak about next generation sequencing. So I know that you're all aware of next generation sequencing, but just to have everyone on the same page, so I'm talking about massively parallel sequencing. So you take a DNA, you fragment your DNA, you make a clonal amplification of these little fragments so that you only amplify, well, ideally just this fragment in one reaction. And then you do a sequencing of all these fragments all at once on the sequencer. So I'm going to introduce to you the Iron Pgm Sequencer very briefly because, yeah, we have only 15 minutes. And then I'm going to present some data on influenza typing using the Iron Pgm. Okay. So almost 2 years ago, Life Technologies acquired Iron Torrent, a company that was founded by Jonathan Rothberg. And he not only founded Iron Torrent, but also 454, Curagen, and Raindance. Very smart guy, actually. So we have offices all over the world, actually. We have global manufacturing sites as well. And this technology was the first one which we call post-light sequencing technology, because we are not using light or fluorescence to identify or to monitor the sequencing, but we're using protons. Okay, so the chip is actually the machine, so the instrument itself is less important. More or less everything is happening on this little semiconductor chip. What happens, I will show you in the next slide. So, but everything, yeah, the whole reaction is taking place on that little piece of metal. It's scalable, so within 1 year we were able to extend or to have a scalability from just 10 million bases to 1 gigabases scale. Depending on what you want to do, you might need a different throughput. So if you're just looking at the tiny viral genomes, you might not need 1 gigabase output, but a smaller one. So it's very flexible in that respect. It's very simple. So the whole procedure, the whole instrument, there are no optics. We don't have a laser, there's no fluorescence. So we just measure the pH or the voltage, which is related to a pH change. And it's very fast. So the sequencing itself can be less than 2 hours, 1.5 hours, not taking into account the library prep, of course, but just the sequencing can be very, very fast. So that's the most technical slide I have today. That shows you how the chip is working. So we have millions of little wells on that chip. And within each well, we have a bead, or ion sphere, we call it. Where's my pointer? So you have these beads, and on these beads you have your template, your DNA fragment, and the sequencing is taking place on these beads. So per bead, 1 fragment. And during the sequencing reaction, protons are generated, and we have a sensing layer here, which is sensitive to the change in the pH, and that is directly transferred into an electronic signal and measured by the instrument. And what we do have is a sequential flow of nucleotides. They are not labeled, so it's just natural nucleotides. And of course, you need to know when you have which nucleotide, and then you get the sequence at the end. That's an overview of the overall workflow. So as I said, you need to construct a library. So if you start with genomic DNA, you need to fragment the DNA. If you have a virus, you might do a PCR amplification. Then you get fragments of a particular region or from the whole genome. So this takes approximately 2 hours to generate this library. And what you then do is the clonal amplification of these fragments. So ideally you would have 1 reaction vessel. It's a water in oil emulsion. and per reaction you would have just one fragment. By a proper dilution, you can achieve a very high or good ratio that you really have one fragment per reaction chamber. So this takes a while, like 4 hours, to do this clonal amplification and then do an enrichment so that you have a high percentage of these reaction vessels where you just have this one fragment. That's what we call enrichment of the beads. So that takes more or less most of the time. The sequencing itself, depending on your output, is really fast, so that's the shortest time. It can be up to 4 hours if you have full human genome, for example. Processing of the data, and this is just the processing on the server, is very fast. What you do afterwards in terms of deeper analysis is a different story. Then, I mean, you can work on that, of course, for much more than just 30 minutes. That's the overall workflow, and this should just show you again, you have the sequencing reaction, you have the processing of the data, and then we have established a very huge community of people who develop data analysis tools, which we call plugins, depending on what you want to do, viral sequencing, bacterial resistance typing, or whatever, so you would need different tools to do the analysis. It's very open, so the community can develop these plugins and we make them available all for free. And we have currently 10,000 members in this community already. And we also have very strong alliance with some third-party software providers. So it's very open in terms of data analysis. We are also currently going to develop a DX instrument. So we are going the CE-IVD path. And as you know, we have the 3500 DX, we have the 7500 DX, and now also the Quant Studio DX. So we have a lot of experience in developing these instruments. And we are currently also developing the Pgm-IVD instrument. So now, what about influenza typing on Pgm? I would like to show you some internal data as well as some collaborator data. Actually, we do have a kit that was developed by our group in Ambien, by our animal health team. This is a custom kit, so you will not find it on our homepage. You have to contact your respective sales rep or us at the booth if you're interested. It contains reaction material for 50 amplifications, so it's the PCR primers, the reverse transcription primer, and the buffers and the enzyme you need to do the amplification of influenza A, for flu A. And it's not only for avian or swine, it's also for the human infecting influenza. So the primers were designed in the conserved region. And you amplify all 8 segments of the influenza A genome. So that's what we have done internally. We have first tested these primers on our CE capillary instruments with Sanger sequencing and compared some of the samples with the Iron Pgm Sequencer. You can see here which types we did sequence. That's the workflow, just to show you again what you have to do if you're going to use the Iron Pgm. So you do a reverse transcription, you do a PCR, you use all of your reverse transcription reaction to do a PCR. You get PCR products with the 8 segments of flu A. So that's optional, it just shows you the agarose gel, then you do an amplicon cleanup, and then you start with the next-gen sequencing preparation. So from here, it's 2 hours again for the library prep, then clonal amplification on the ion spheres and enrichment. Sequencing in that case is 3 hours because we have longer fragments we are sequencing. We did use 200 base pair fragments. And then we did data analysis. That was also developed in-house, this analysis pipeline. I'm coming back to that because collaborators have also developed their own analysis workflows. So we mapped to all available reference sequences. So that's the first step you do with the output material. Then we looked at the coverage, and we took only the sequences or the fragments which showed good mapping to the reference, plus had a high coverage in terms of reads, because per base you get like hundreds of reads. That's what we call coverage. And from that, we generated a consensus sequence, which is then uploaded to reference database for further analysis. I have the detailed results with me. If you're interested to see them, for the sake of time, I've summarized them here. We compared the data with some published primers, which are very common in the community. Comparing both protocols, we could call all genotypes, so we got consistent results between the two different protocols. We could see that the shorter segments have a higher coverage. If you know PCR, you know that the shorter fragments are, you get a higher or better amplification. That's that's common to PCR, so you would expect that you also have a higher coverage. So in the next step in optimization of this protocol, we would try to balance that you can also, that you get an even coverage for all segments. We also mapped the data to the host genome, so for to the dog genome and to the chicken, and we got a very, very low mapping to the host. That was much better than with the collaborators or the published primers. It was also highly sensitive because we did do a dilution series, and as I said, I have all the data with me and we can have a look at that. And we could go down to 200 copies of RNA, so we did an RNA dilution, and we could still call the right genotype. And now I would like to speak briefly about my project. I'm coordinating a global network, which we call the FLU network, with currently 6 institutes. One is missing here. So we have the SMI in Stockholm, which is closely connected to the European CDC. We have the ISS in Rome. We have the CDC in North America, in Atlanta. We have another veterinary institute in Iowa, in Ames. And we have the J. Craig Venter Institute in Rockville. So what do? We have connected these customers to define with us the standard operating procedures for flu typing, because this new technology is so revolutionizing that we need new standards. And how do we look at the data? How do we store the data? How do we interpret the data? So we are working together, and that's a very, very nice interaction, I have to say. And the 6th institute is the China CDC. They just joined recently. So we are working on a protocol. We will share and discuss results, and finally, of course, we want to publish the results. And one of the members is going to speak in 4 weeks at a PGM user meeting where he will present the results. If you're interested in that, just come to the booth. We want to broaden the network in the next step, so I'm happy to get your input, your requirements. And that's actually my last slide. That's now data from the CDC in Atlanta, and I'm very happy that I can present it here. They didn't use our kit, so they did use their own primers for reverse transcription and PCR, and they did run their own proof of concept study on the Iron Pgm, and here are their results. So they did do multiplexing, so they came out with a 12-plex, so 12 samples on one chip using barcodes. And they saw that with a coverage of 25 to 50-fold, which is rather low, you would say, they could still call the right genotypes. And that's what I meant in terms of throughput. If that's sufficient to get a consensus sequence, then you don't need a high output or a high coverage per sample. So you can still do a correct genotype. And they showed, because it was a proof of concept study, yes, they can use the Iron Pgm to successfully do influenza sequencing using their own primer set. And what I found interesting is that they really think about a paradigm shift